To understand the female reproductive system, we first have to look at the baseline established by the male reproductive tract. That system is highly optimized for two straightforward objectives: the continuous production of sperm cells and the physical delivery of those cells into an external environment. Knowing that, a natural comparative question arises. Between the male and female biological frameworks, which system requires a higher degree of structural complexity to achieve its goals? Placing these two blueprints side by side highlights the divergence in functional requirements. While both systems are responsible for gamete production, the mechanical load on each structure varies significantly. As the schematic fills in on the right, we can see the female reproductive tract carries a significantly heavier operational load. First, it has to produce a gamete and actively facilitate the precise microscale mechanics of fertilization. Second, the system must expand into a massive, scalable environment capable of supporting the complete morphological development of a human baby over 9 months. To map how this dual-purpose architecture actually works, we need to build the system from the ground up using a first principles approach. We can do this by tracing the exact spatial journey of the female gamete, starting from its absolute origin point and following it all the way to the central site of gestation. What emerges is an end-to-end biological framework strictly engineered to initiate, secure, and physically sustain human life. The journey begins at the specific spatial origin point of the female gamete. These are the ovaries, two bilateral, oval-shaped structures positioned on either side of the lower abdomen. Their primary role is the maturation and release of the egg cell, or ovum. Unlike male sperm, which are equipped with flagella that allow them to swim through fluid environments, the ovum has a severe physical limitation. This 3D model isolates the core mechanical constraint. The ovum is entirely non-motile. Once it leaves the surface of the ovary, it has no biological means to propel itself forward. Making matters more difficult, there is a distinct anatomical anomaly here. The ovaries do not physically connect directly to the adjacent transport tubes. There is an empty spatial gap between them. To bridge this gap and prevent the egg from simply drifting into the abdominal cavity, the system relies on a specialized engineering solution. Attached to the ends of the transport tubes are the fimbriae, a series of distinct, finger-like projections. These fimbriae actively reach out across the void. They perform a continuous, rhythmic sweeping motion to capture the released ovum. This sweeping activity creates a fluid current strong enough to secure the static cell and physically push it into the opening of the adjoining corridor. Without this continuous mechanical force provided by the fimbriae, the non-motile ovum would be stranded upon release, rendering the rest of the reproductive cycle physically impossible. Once the ovum is successfully pushed past the fimbriae, it enters the next major structural pathway. This bilateral transit corridor consists of the fallopian tubes. The fallopian tubes provide the specific structural environment required for the transition from individual gametes to a multi-celled organism. As we look at the cross-section of the fallopian tube, we see its critical active function. This environment is the dedicated site where fertilization occurs. It is here, within the narrow confines of the tube, that millions of sperm swim upstream to intercept the slowly drifting, non-motile ovum. The successful entry of the sperm cell into the ovum terminates the transport phase and initiates the process of cellular fusion. As the genetic material combines, the single ovum cell becomes a zygote. This newly formed zygote does not remain static. It begins a distinct behavioral change as it prepares to leave the corridor. It initiates rapid cellular division. By the time it drifts further down the fallopian tube, it has already transformed into a multi-celled embryo. The fallopian tubes operate as a highly regulated intersection point, engineered specifically to force the collision of two distinct physiological pathways to initiate life. The dividing embryo eventually reaches the end of the fallopian tube and exits its narrow confines. It arrives at the central destination of the entire tract, a large, muscular, pear-shaped cavity. This primary structure is the uterus, commonly referred to as the womb. This diagram outlines the exclusive operational purpose of the uterus. It provides a scalable environment designed for long-term fetal growth. The arriving embryo embeds itself into the uterine wall, securing a nutrient supply where it will remain until morphological development is complete. Beneath the main cavity of the uterus, the anatomy funnels down into a narrow lower exit. This exterior connecting passage is the vagina. This passage must satisfy a unique bidirectional mechanical requirement. Inwardly, it acts as the intake point for receiving sperm into the female body. Conversely, it also performs a demanding outward function, serving as the exit passage for the baby during childbirth. This lower tract acts as the initial entry gateway to trigger the possibility of fertilization, and the final exit gateway to culminate the process of gestation. Stepping back, we can now view the complete architectural model of the female reproductive system. The sequential pathway begins with gamete production at the ovary. Next is the physical capture of the non-motile cell, performed by the sweeping of the fimbriae. Transit follows, providing the conditions for microscale fertilization within the fallopian tubes. Then the embryo anchors inside the uterus, initiating the expansion required for macroscale development. Finally, the system connects to the exterior through the dual-purpose vaginal canal. Returning to our initial anatomical comparison, the structural reality of the female framework is clear. The female reproductive architecture is defined by a higher degree of complexity than the male counterpart, driven by strict biological necessity. The system must constantly balance the fragile logistics of cellular transport against the prolonged physical demands of gestation. By integrating these distinct mechanical and biological zones, the system provides the specific conditions necessary for a single cell to become a viable human being.